Hello students, so welcome back to the channel. In this video we are going to study about chromosomes. Now you know very well in the previous video we have studied about nucleus. Okay, and in this nucleus if you remember we discussed that nucleus is having chromatin. So nucleus is seen during the time when the cell is not dividing, means interphase. And in the interphase nucleus you are finding chromatin is present. This is a thread-like structure called chromatin, which will condense to form chromosome at the time of cell division. The chromatin condense to form chromosome. And at the time of cell division, in place of nucleus, you are finding chromosomes. So today our topic is, we'll be discussing about the chromosomes. So first of all, see, they are known as physical basis of inheritance. So chromosomes are physical basis of inheritance, and genes means DNA. Let me just write it. And the DNA is a chemical basis of inheritance. DNA is the chemical basis of inheritance. So first of all, this is clear which is physical basis and which is chemical basis of inheritance. The chromosome is a physical basis of inheritance, and chromosomes are made up of DNA and protein, in which DNA is a genetic material. That is why we say DNA is a chemical basis of inheritance. Now, if we see the discovery, we find it is discovered by, chromosomes were discovered by Hofmeister, and they were discovered in the pollen mother cell of Tridescantia. This is a plant. In its the pollen mother cells, there they discovered it. But the term chromosome, this term chromosome was given by Waldier. Because your chromo word means color, okay, and zome means body. So they were colored body because they can take the stain with the basic dye. They were able to stain with the basic dye, and the dye was Janus green. And Sutton and Bowery has given chromosome theory of inheritance. If you remember in the genetics we study, Mendel is known as father of inheritance. He is the one who has given all the laws how the character are transmitted from parents to progeny. But he was using word factor, कि parents are giving to their children factor. Okay, so he was not able to know the physical proof of that factor. So physical proof of the factor was given by Sutton and Bowery, and they told Mendel factors are nothing, chromosomes. So he gave the chromosome theory of inheritance. So this is with related to the discovery of the chromosomes. Now let us see the number of the chromosomes. Now see, few facts are there with respect to the number of chromosomes. Two types of organs are there. One which are having main body as haploid body, which we generally see in the plant and in the lower plants like algae. So in that also we are finding there is minimum number of chromosome, n is equal to 2. So this is seen in Porphyra, and Porphyra is a member of red algae. And maximum number can go to n is equal to 592. This many chromosomes can be present in another algae. This is also an algae, that is natrium. So these are the examples of the haploid number of the chromosomes, means the minimum number of the chromosomes and maximum number of chromosomes in the haploid organism. Another example is in diploid organism. We have taken the case only from the plant. If you see here, minimum number of the chromosome is 2n is equal to 4 in Haplopappus gracilis. This is an example of angiosperm. And maximum number in the entire plant kingdom, you will find 2n is equal to 1 is present in the Ophioglossum. That is 1, 2, 6, 2. These many chromosomes are present, and Ophioglossum is fern. So here I just want to mention that human has 23 pairs of chromosomes. 23 pairs of chromosomes. It means total chromosomes are 46. 46 chromosomes, out of which 44 are autosomes. And the last chromosome, that is XX in female or XY in male. This is known as sex chromosome, also known as allosomes. The sex chromosomes are also known as allosome. So I hope with respect to the number is clear. Of chromosome. To see carefully. First of all, you see this chromosome only during the time of cell division. So this is primary constriction, and these are the two chromatids. Check my diagram carefully. So see carefully, this is DNA. So this is full one chromatid. I'm using blue color to indicate one DNA is present and it is making one chromatid. Okay. I'm using another color just to understand only. So this is the one which is replicated at the time of the interphase. And now this is your second chromatid. So these two chromatids are attached to at one point. This is known as primary constriction. Primary constriction is also known as centromere. Primary constriction is also known as centromere. And these two are known as sister chromatid. These are known as sister chromatid. Is it clear? So one chromosome I have drawn which is having one primary constriction and two sister chromatid. End of the chromatids, check the diagram, end of the chromosomes means chromatid, this is a structure. This is known as telomere. So it is, see carefully, these are the telomeres. And on either side of the centromere, you're finding this proteinaceous disc-like structure. This proteinaceous disc-like structure, which is known as kinetochore. Is it clear? So we can say, right now I've drawn only one chromosome. So we can say one chromosome is having two chromatid. It has two chromatid and they are known as sister chromatid. It is having two DNA because one chromatid means one DNA, two chromatid means two DNA, but it is having one centromere. It is having two kinetochore. And it is having four telomere. These are the things you should know when you are solving the questions. And this is the structure which you can see best phase. Best phase to study the structure of chromosome is always metaphase because they have reached to the maximum condensation. So this is your metaphase chromosome. Is it clear? This is the best phase to study the chromosome structure. Now, some chromosomes may have, apart from this primary constriction, some may have secondary constriction also. So let me explain you the diagram with the secondary constriction. So this is primary constriction, okay? And I'm now drawing, this is secondary constriction which I have drawn. Two chromatid only, a chromosome with two chromatid, okay? But if you see here, apart from primary constriction, it is also having another constriction that is known as secondary constriction. Let me just draw this thing after your DNA. So this area, this is known as secondary constriction. And as a result, this structure which is made, this structure is like a bulb-like, knob-like structure. So this is known as satellite. Okay, telomere will be there. Telomere will be total 4 only. It will be having that these tips are known as telomere, okay? And you will be having this kinetochore also. This is the proteinaceous disc kinetochore is present. Now, because of this satellite, this chromosome is known as set chromosome. Now, these are the set chromosomes. Because of this, they are having secondary constriction has NOR region. The secondary constriction has NOR region. NOR region means nucleolar organizer region. Means this is the one region which is going to make nucleolus at the telophase. So I'm just writing it. This region make nucleolus at the telophase. At the time of the telophase, they will make nucleolus. And there are 5 pairs of set chromosomes are present. Okay, 5 pairs of set chromosomes are there in human. That is chromosome number 13, 14, 15, 21, and 22. These are the 5 pairs of the chromosomes which are having secondary constriction. So they are known as set chromosome. Please check your NCERT diagram. Then you will be able to understand this concept more clearly. Okay. Now, depending upon the position of the centromere, right now in this diagram, I have drawn the centromere exactly in the middle of the chromatid. Okay. Now, centromere can change. It is not necessary exactly it can be in the center. It can be shifted a little bit away from the center also. So, depending on the position of centromere, we'll be classifying the chromosomes into 4 categories. Now, let us see. On the position of centromere, let me just draw the diagram. This is your a chromosome with 2 chromatid, and if you check the diagram, this upper arm, this is upper arm, okay, means above the centromere, that is known as upper arm, and this is lower arm. So both are equal. So arms are equal. If arms are equal, and because why the arms are equal? Because the centromere is exactly in the middle. So we say such chromosome as metacentric chromosome. Is it clear? Metacentric chromosomes are those chromosomes which are having equal arms. We call such as isobranchial chromosomes. Is it clear? Isobranchial chromosome means equal arm. Now check the next diagram. Check carefully. The centromere is not exactly in the middle. So as a result, upper arm has become short and lower arm is long. So 2 unequal arms are created. Short arm is also known as, please, this is again PMT question. Short arm is known as PR. It is known as PR. And long arm is known as QR. Okay, long arm is known as QR. Learning tips is this way. You can learn this way. Q stands for queen. The queen is obviously big lady. So Q means it's a long arm. And P you can remember like a peon. Peon is a short person. Okay. So P for the peon, you can remember the short arm is known as P arm and long arm is known as Q arm. So now if you see the centromere is not exactly in the middle, it is shifted away from the middle. So we will say submetacentric. Metacentric chromosome. Now, if suppose it is trying to reach to the apex and as a result, one very short arm and one very long arm. Okay. So this is your very short arm and this is your very long arm. So it is trying to reach to the apex, but it has not reached to the apex. So we will say this is acrocentric chromosome. And if suppose the centromere has reached to the tip, it has reached to the tip with very small upper arm, which is actually you can negligible also, ignore it also. So we will say such chromosomes are known as telocentric chromosome. So depending upon the position of centromere, your chromosomes are of 4 types: metacentric, submetacentric, acrocentric, and telocentric. Now see beta, we have already discussed in the video also, that interface nucleus will have the chromatin. Is it clear beta? But during the time of cell division, you will find chromosome in place of nucleus. And chromatin contain DNA and some basic protein, that is histone, and some acidic protein which are non-histone protein, as well as RNA. So all this we have already covered in the nucleus video. You can check it also. Okay. A single human cell. And if we see that our DNA is nearly 2 meter long, 2.2 meter long, the DNA. Reason is that we are having nearly 6.6 into 10 raised to power 9 base pair. These many base pairs are present. Let me write for you so that you can understand it. Now see beta, we already discussed that interface nucleus has nucleoprotein fiber called chromatin. Okay, but at the time of cell division, means in the prophase and slowly in the metaphase, by the time they reach to the metaphase, cell will show chromosome in place of nucleus. So instead of nucleus, you will find chromosomes. And what is chromatin made up of? It is made up of DNA. It is made up of basic protein to pack the DNA. And for this, you can do one thing. You can go and check one video is there in the playlist, that is a packing of the DNA in eukaryotes. You can check it, that you will understand how they are making use of histone proteins to pack it. Okay. Apart from this, they are using non-histone protein also and RNA. This much is clear. Human cell is having nearly 2 meter long thread of DNA. Okay, so DNA of human is nearly 2 meter long, which is present in nearly 46 chromosomes. So I told you human has nearly 46 chromosomes, is 23 pairs, out of which 44 chromosomes, out of 46, 44 chromosomes are autosomes, and only the last pair is sex chromosome, also known as allosome. Now let me just tell you how we are having the DNA 2 meter long. Just see better, just give me 1 minute. See carefully. Human genome. First of all, human DNA, we are talking about diploid, okay, 2n content, is made up of nearly 6.6 into 10 raised to power 9 base pair. These many base pairs are present in human. Is it clear? And the distance between one base pair, distance between one base pair is 0.34 nanometer. So if you multiply it, total DNA, if you multiply 6.6 into 10 raised to power 9 base pair, if you multiply it with 0.034 nanometer, your answer will come nearly 2.2 meter long. Clear beta? So that is why this calculation, this topic is again, we'll be discussing in the biomolecule, but in the playlist again one video is there with respect to new bonds in the DNA. You can go and check it. It will be helpful for you. So up till here, is it clear? So now see, every chromosome is having primary constriction. Okay, check the diagram carefully now. You're finding the chromosomes are having primary constriction, that is known as centromere, on the side of which disc-shaped structure. Please, again, it's a question. What is the shape of the kinetochore? The kinetochore is disc-shaped, and you can see here the diagram is showing it. Okay, kinetochore is a disc-like structure which is present on the either side of the centromere. Based on the position of centromere, the chromosomes are classified into 4 types. If you see the diagram, first diagram is metacentric. They are exactly in the middle, centromere. And in the second diagram, it is shifted. As a result, one short arm and one long arm is coming. Clear beta? Third is your acrocentric and fourth is your telocentric. So check carefully in the theory. Metacentric chromosome has middle centromere making 2 equal arms. That is why I wrote isobranchial, equal arms. Submetacentric chromosome has centromere slightly away from the middle, resulting in one short arm and one long arm. And in case of acro, the centromere is very close to end, making one very short and one very long arm. But in telocentric, it has reached to the tip, terminal. Is it clear? Now, if you see this diagram carefully, you can notice in this diagram, secondary constriction is also there. Analyze the diagram better. Secondary constriction is there. And because of secondary constriction, you can see the bulb-like structure is made that is known as satellite. Is it clear? So see here, sometimes a few chromosomes, this is also non-staining part. Primary constriction is also non-staining. Even secondary constriction is also non-staining. Okay, and it is at a constant location, and it gives an appearance of a small fragment called satellite. So such chromosomes are known as set chromosomes. So I hope this topic is clear to you. Okay, thanks for watching.